Już? Co? How? Okay. Fine? Yes, I know it's fine. Okay. So sorry. So welcome everyone to the webinar series organized by the Polish National Agency for Academic Exchange called Ready, Study, Go, Poland for Students in Poland. So now let me start from my presentation. Thank you for invitation. This is this is most important. And let me tell you today about the Polish discovery. Let's say it's Polish discovery about the about the first Polish diagnostic kit for SARS-CoV-2 detection. And what I'm going to tell you today is the story about the first Polish diagnostic kit, of course, but the story about the efforts of many people, really many people, to make this idea to came to the reality. And hopefully I won't waste your time. So let's start from the beginning. So let me introduce myself and let me introduce my team. So usually, now I can say usually, I'm the head of the Department of Molecular Neuro-Oncology in the Institute of Bioorganic Chemistry, Polish Academy of Sciences in Poznań. And I'm happy to work with these awesome people, which you can see on the photos. And what is our main research area? Our research area are the brain tumors, especially glioblastoma multiforme, which is the most devastating brain tumor. Is the most common and aggressive brain tumor with the prognosis of 12 to 15 months of median survival. The standard treatment comprises surgical resection, which is sometimes followed by chemotherapy and radiotherapy. The malignancy of the tumor is divided into 4 grades, from 1 to 4, where the 4th grade is the most malignant. This tumor is characterized by really invasive capacity, which makes actually the surgical resection impossible. What adheres to the radiotherapy and chemotherapy resistance. So this tumor very often we can observe very often the recurrence of this tumor in all patients within 2 years after completion of the treatment. So what we are interested in during our research, what we are looking for, is actually the development and progression and recurrence of the tumor. So we are really interested in the mechanism of this all issues, and for us, RNA is the most important in this whole story. So we are looking for diagnostic, prognostic, and therapeutic molecules, especially RNA molecules. We are looking for mechanism of action with involvement of these RNA molecules, and what we are also would like to implement in our research is the new research models, quite sophisticated models, which really will allow us to find more appropriate mechanism and more reliable will be the more reliable models to this which we use nowadays. So let's look our normal life in our laboratory. So we spend our time in the lab as you can see on the pictures, we spend the time in working with the cell cultures, working with many different molecular biology methods. But our life changed completely in March 16th because of the coronavirus epidemic. And with this, I would like to tell you how the story began, actually, what we did during last month to achieve the first Polish kit for COV-2 detection. So, as I said, it started on Monday, March 16. That was actually our first day of our home office. So we were sitting at homes. We were in the morning. We had a Skype meeting, and we discussed about how many papers will we prepare during this time. What will we complete? Where will we send these papers? So we were really ready to sit at home and write the papers. But around 10:30 p.m., our director called me and he said, "You know what? Director of the epidemiological sanitary unit called me this evening and he said that they are completely stuck with the samples from the patient. They can't diagnose the patient anymore because they don't have the people to do it." So he said, "You know what? It's really a war. We have to be involved in this because we are a public institution. It's our duty. We need to do it." And he asked me how many people I can somehow involve in this project. So I said, "I don't know. I have to ask them." So we start to call to each other, and around the midnight, we had the decision that whole our department will be involved in the project. So next day we had a meeting in the institute. So there were maybe 15 people or so, but people came again and again, and more people and more people and more people. And suddenly we realized that we had almost 50 people, volunteers, who really wanted to be involved in the first only in diagnostics. So we had the meeting around 10:11. Around 5 o'clock, we had already 90 samples from our sanitary unit we knew that it was only small part of the samples, because we knew that they had already 800 samples freezed in the freezer, and they had no capacity to diagnose all the samples. So we started to diagnose it with these 90 samples. But we knew from the sanitary unit that there is not only the need to diagnose the samples, but they said that there is a lack of isolation kit, RNA isolation kits, that there is the lack of detection kits. And we also knew that the kit which is used already for diagnostics in Poland is relatively expensive. So from the first day we knew that apart from the diagnostics, which was our, let's say, main goal, there will be also the optimization or developing our kit, Polish kit, which will be used in diagnostics units in Poland. So on this slide, you have the situation from already Tuesday also. As you can see on the map, our institute is located in 3 locations in Poznań. So our lab is located in University campus. So I've marked it with the circle. And in order to somehow help in our main location, we took everything what we could take from our lab. So I mean the pipettes, tips, many different things, which we knew that will be helpful for this project. So on the picture is Konrad, is one of our PhD students who took all of this stuff from the lab and he brought to our main location. He also took from our lab qPCR machine, which I will explain you later, to make it somehow useful in the project, in COVID-19 project. So our ultimate goal, as I said, was COVID-19 diagnostics. We knew after 2 days of 2 days of our work that we need to do it, and we should help to our diagnostic unit. Okay, so the next goal which we planned to do it was the Polish test design. So we started to look at many different tests which were available on the Polish market, and also in the German, in French, or even American market, and we decided to do our own test. How this whole procedure looks like. So what should we start with to do all this diagnosis? So first you have to take the biological sample. So the sample is basically taken from the throat. Then the samples should be collected in the units where the samples can be collected, because this is potentially infectious material, so it has to be stored in an appropriate laboratories. Then this material has to be, from this material we extract RNA. Then the diagnosis is done through RT-qPCR, and finally we have test results, which is visible as the curve on the picture. So before I come to the test itself, since I don't know whether you are all familiar with these molecular biology terms, I would like to give you some short vocabulary. And let's start from RNA, which I've mentioned before. This is the main molecule which are interested in. It's apart from DNA, the second most important nucleic acid in our cells. And cellular organisms use messenger RNA, mRNA, to convey genetic information using the nitrogenous bases of guanine, uracil, adenine, and cytosine, denoted by the letters G, U, A, and C, which you can see also at the slide. The direct synthesis of specific proteins, and what is important in terms of our project is that the many viruses, including SARS-CoV-2, encode their genetic information using an RNA genome. So the next term which I would like to show you is a nucleic acid sequence, which is just simple base pairing base pairing order, which is counted from 5' to 3' end of the molecule. It's, as you can see, is it's written in the letters, as I mentioned before, G, U, A, and C, and that's the nucleic acid sequence. Then, what is actually crucial for this analysis and for our set and detection kit and for diagnosis of SARS-CoV-2 is the real-time polymerase chain reaction, which is called real-time PCR. It's known as quantitative polymerase chain reaction, and is one of the most popular technique of molecular biology based on the polymerase chain reaction amplification. So DNA fragment is amplificated through the polymerase, and the results are obtained in the real time of the amplification, and they are measured in the cycles of the reaction. Usually we use 40 cycles of the reaction, and I also will show you how it looks like later. So two common methods for the detection of PCR products in real time are used, and first there is non-specific fluorescent dyes that intercalate with any double-stranded DNA, or what is the most important in our case, it is the sequence-specific DNA probes consisting of oligonucleotides that are labeled with fluorescent reporter, which permits detection only after hybridization of the probe with its complementary sequence. What you should also know, and you should also keep in mind, is the term of primers, which are short DNA fragments complementary to the 5' end of each of the sense and anti-sense strands of DNA target PCR reaction. These are crucial for amplification of the target. Then cDNA, which is complementary DNA generated by reverse transcription of mRNA. So when RNA is the genetic material, you have to convert it into cDNA to amplify it later. And what is also important in our case is the fluorescent reporter probe method, which we used for our detection kit, and it's based on we use in this kit the special probes, which are somehow devoted to detect only the DNA containing the sequence complementary to this probe. So this probe, fluorescent reporter, this probe have attached the fluorescent reported at one end and a quencher of fluorescence at the opposite end of the probe. So from the 5' end, you have, for example, FAM, and on the 5' end, you have BHQ quencher. So how it works? As you can see on the picture, it attaches to the sequence. What makes all of this amplification really specific and really sensitive and really accurate, I would say. So this fluorescence is detected and measured in a real-time PCR machine, in qRT-PCR method, in the appropriate detection channel. So when we think about the diagnosis with actually genetic test, as it is in terms of qPCR based test, we need to look at the sequence of the virus. So what we started with to really design the specific primers. So as I said at the beginning, SARS-CoV-2 is a positive sense single-stranded RNA virus. So it means RNA is the genetic material for this virus. If you can please look at this at the picture, where you can see this genetic this genome, virus genome. So it's somehow ordered into, it's quite, I would say, quite small genome. And it codes very few proteins which are really necessary for the virus. And what we look at the beginning was ORF1 and ORF1A and ORF1B sequences. And we designed actually the primers according to this region. We also checked in terms of this region many different tests, as I said, available on the Polish market, and not only Polish market. And we found that this region is not only very conservative for this for the virus, but it's also very often used in many different tests. So we decided to start with our test with very simple version of the test. And we decided to use the primers and the probe, which were designed especially to ORF1A and ORF1B sequences. Then, as I said before, we had to use and we had to design specific primers and also specific probes. So, as you can see on the left-hand side, Tagman detection method illustrates the usage of the primers and usage of the probes. So for our gene of interest, which was, as I said, ORF1AB, which will be, and it is actually confirmatory testing for COVID-19, we use Tagman probes, which were which have dyes at the 5' end, as I said, FAM, and at 3' end, it was BHQ. For the second gene of interest, which was R18S RNA, which was our confirmatory testing for an appropriate biological sample handling, we used HEX at 5' end and BHQ again at 3' end. So with this design, we basically have the general basic scheme for the diagnostic test setup. I would like to explain you why we choose R18S RNA as the confirmatory testing for the biological sample. So, as I said, we started from the diagnostics in our institute. So we had many different samples, many samples which we diagnosed as positive and also negative. And we had no doubt that these positives tests are really positive. But we couldn't say the same about the negative probes, because we had no information, not at all, how the samples are taken, how they are kept, whether this RNA material is still really present in the sample or not. That's why we decided, since the biological sample is taken from the throat, that's why we decided to use R18S RNA as the which is the human gene, as the test for this biological sample appropriate handling. So, what was the biggest challenges in our COVID-19 story? So, in terms of diagnostics, we faced with the RNA isolation. So we had to we still have to know, and we still have to have really fast, pure, and safe methods for RNA isolation. and with the amount of the samples, it will be good that it will be also cheap. So for this diagnostics, we also need a reliable PCR analysis. So we need also the test, which will be accurate, which will be specific, which will with any doubts will detect RNA from the virus. So in the parallel, we also were working on the test. We still work on the test. So as I said, we focused on the high efficiency, of the high accuracy and purity of the items which were used in the test, and high specificity and selectivity. On this graph, so the work on this test, it's not really impressive. There are many pipetting steps. There are many PCR runs. So I can show you on this slide. I will show you the example of our results. On the first graph, you have the positive patient, because as you see on the curve, the amplification starts around 24 cycle. So as I said in vocabulary, that RT qPCR procedure is somehow measured in the cycles. So where you will have patient which is detected in 24th cycle, it means that the RNA of the virus is really present at the high level. On the middle graph, you have the well, I can only add. So when you look at the small description on the graph, on the first graph, you have the channel which is taken as FAM. So we somehow we prove that it's observed in the right channel, because for this detection, for this sequence, we used probe which was labeled with FAM at 5' end. At the middle graph, you have the R18S RNA, which was our reference gene. And this reference gene is even more abundant in the sample than RNA of virus. And this result is observed in the other channel, which is hex channel. So we knew that this patient was for sure positive. When you look at the table, there is a description and interpretation of this results. So when you will have two pluses from FAM channel and hex channel, you will have for 100% a positive patient. When you have plus result from FAM channel but minus from hex channel, still you have to consider that your patient is positive. When you will obtain the result on FAM channel, which is minus, and on hex channel, which is plus, it means that the biological sample was taken in appropriate way. So for sure we have the RNA in the sample, but we don't have the RNA of the virus. So the patient is diagnosed as the negative. But when you obtain two minuses in the channels, in both channels, in our interpretation, this patient needs to be analyzed second time. So most probably we would have to take the another sample, biological sample, or we have to again isolate RNA, make qPCR, and obtain the results. So I've put our efforts, our work, I put on the timeline, which as as I see now, it looks almost impossible because we started a month ago. So as I said, on March 16, we were sitting at home office. At March 17, we arrived to the institute again. We started to diagnose the patient. On March 18, we first we synthesized. We somehow designed and we synthesized the primers and probes in the most appropriate way to the sequence which we found. Next day, we had to contact to the local, to the Polish, actually, not only distributors, but Polish producers of the qPCR reagents for qPCR buffers. And then on March 22nd, we already had COVID-19 gene, which was ORF1AB. We had this gene approved as the working gene in our hands, in our kit. Of course, during all of this time, during these days which are really unmarked, we optimized the kit, we optimized the conditions, we optimized the concentrations of these primers and probes, the temperatures of the reaction, to make the second gene, gene also be approved as the confirmatory gene of the biological sample. In the meantime, whole diagnostics was ongoing. So on March 27th, we have already 1,000 samples, which were diagnosed in our institute, and 3,000 samples we had analyzed. We had analyzed on April 7th. In the meantime, we have to validate the test in the external units. So our test was sent to our sanitary unit in Poznań, was also sent to Warsaw, and 2 independent validations prove that our test really works with the patients. During this time, also, as you can see on March, almost on the last day of March, we sent the application to the Ministry of Science and Higher Education, and we asked for money for production of the tests and for the optimization of the next version of the tests. Since we are research unit, not the company, not the pharmacy, we had to contact with the pharmacy company in order to obtain the necessary certificates to somehow make this product to be useful on the market. So I mean, only certified tests, only certified kits can be used in our diagnostics unit, even in pandemic situation. So we contacted to the company. We prepared together with them so-called IVD application, which was, as I said, very important to somehow put this product into the market. Then next day, we found out that Ministry of Science and Higher Education granted our application. And then a few days ago, we produced 800 tests to be used as a demo kits. And now I can tell you that next week we will start with production of the first 10,000 of the tests. So I can show you on the few pictures how this work looks like. So this is the isolation laboratory. Here you have again RNA isolation work. Here you have pipetting, so preparation for qPCR analysis work. qPCR, this is how this machine looks like. It's not very impressive, as I said. The work with the test, it's not very impressive. It just or not only just hard work. There are many hours in the lab, many hours with the pipetting, many hours with the re-pipetting. So yeah, this is again optimization, qPCR machine, and finally I can show you for the first time our product. This is Medipan COVID test. This is, I would say, this is our pride now. It will be, as I said, it will be produced next week, but this is a demo kit with all of reagents inside, with all of the necessary labels. And this is because of the work of these people. So as I said at the beginning, there was 54 people. The list of volunteers is 54 people. Many of them are involved in RNA isolation. Many of them are also involved in diagnosis at the last step, which means at qPCR experiments. Also, few of them are involved in the test design and the test design and production now. So why or how it was possible that this work went so quickly? So I would say director of the sanitary unit called to the right institute, because we all in the institute have the experience with RNA. Many of us are somehow connected to virology, but the rest, I would say the majority of us, work with totally different topics. But RNA is very often in the center of, is the focus of our interest. We use this technique, which is qRT-PCR or RNA isolation, almost every day. So we are really familiar with this, really familiar with these methods. That's why we could optimize whole procedure very quickly. When we realized that there is a lack of the kits, for example, for RNA isolation, we just ran to the laboratory and we looked around and we said, okay, we had this kit, we have this kit, maybe we can use this or that one. That's why we were also able to very quickly optimize the procedure of the RNA isolation. The same situation was with the qPCR and with the qPCR mixtures. So I would say, thanks to these people, we have this product, and thanks to these people, all of this was possible so quickly. Yeah, and that's it, I think. That's it, what I would like to tell you today. Hopefully, you now know how we worked with this with so-called Polish discovery. I wouldn't say maybe Polish discovery because we really relied on the literature, on the existing kits. But this, all of this allowed us to make it very quickly, and the kit now will be available on the Polish market, will be much more cheaper than the kits which are now used in the diagnostics units. So hopefully we somehow will help with COVID-19 epidemic. Thank you very much for your attention. And I would like to tell you at the end, please follow the series of the next webinars called Ready, Study, Go Poland for Students in Poland, organized by the Polish National Agency for Academic Exchange. Invitations and more information about future webinars you will find on the website gopoland.pl and on Ready, Study, Go Poland social media fan page or Facebook or Instagram. So please follow these channels. Thank you very much again. So if you have any questions, please feel free. I will try to answer. Okay, so I can see the question, which is what is more or less the percentage of accuracy of the test? So we counted our accuracy of the test. We counted as 99%. So I can't tell you what is the less percentage of the accuracy of the test, but the test is really specific, really sensitive, and it really detects the even small amount of the RNA, because when you refer to the cycles of the qPCR experiment, as I showed you only one example where the SARS-CoV-2 was detected at really high level, but we also have the samples which are detected at the really late, in the really late cycles, which is like 32, for example, or even 36. So we assume that there is really a small amount of genetic material of the virus, but it's still detectable. Okay, whether this test is approved by World Health Organization. Okay, so since we put we had to apply for IVD certificate. This IVD certificate allowed us to use the kit in the Polish market. With this certificate, the kit is also it can be used abroad. It can be used in Germany. It can be used in France. It can be used in the US. But always, it's not only about of our kit. It's all about all medical products. With IVD from one country, you have to somehow introduce the product into the given market. So, for example, in US, with our kit, local distributor in US would have to also apply for the permission, but it's always very simple permission since the kit is already certificated by IVD from another country. Okay, so actually When do you think the vaccine will be invented? So medication. So I think that the work with vaccine really go fast. Some medication, so I'm hoping that it will be soon. I can't tell. I don't know exactly how it works. I'm not virologist, so I don't know how it works. On which level this work is. But I know that there are many efforts in many countries to make the vaccine, and I think it is really possible. It's really possible to have it soon. But it takes a time, always. It takes a time with the production, it takes a time with the testing, so it will take a time. So some medication, it's already on the market, a medication. It was registered according to the other illness. I think it was used in pneumonia, but now it's used as the complementary medication for COVID-19 patient. Okay, the next question is: are you considering expanding your production amount after the 10th of next week? Yes, for sure. The first week we are going to, in the first week we are going to produce 10,000, but the money which we obtain from the Ministry of Science and Higher Education allowed us to produce 150,000 of tests. So till the middle of June, for sure, we will produce all of these tests. Do we will have the possibility to buy the tests from the hospitals from other countries? That's why we also, this is another reason why we combine our efforts with the pharmacy company. With the help of the company, we could somehow expand on different markets. So we will be able, after the 150,000 tests where we already have the money, we will be able still produce this test. We will be able to still improve the test, and we will be able to sell the test abroad. I can see that there will be next question. You know what? I would like to avoid this answer of this question because I'm not virologist, as I said at the beginning. What is the mechanism of action of COVID-19? There are many information in the internet, but what is most important in the professional journals. So I wouldn't like to answer this question because I would repeat only what I read in the internet. So all of you know what are the symptoms of COVID-19. What is the exact mechanism of action? I'm afraid that we don't know exactly yet. Hopefully. So Sebastian asked about the difference in the price between your test and the others used in Poland. Is because of everything is made locally, or more because of it's both actually. So we optimized the primers and the probe. The probe is actually the most expensive item in the kit. So we optimized the test to really make this concentration as low as only possible. So that was the one part of the lowering of the price. And the second, which is extremely important, is that we combine our efforts only with the Polish companies, which are really many times less expensive than the same products from the different companies, for example, from Germany. So both the concentrations of the primers and the probes, and also because of the production on the Polish market. How many per week? So our capacity is like now it's 20,000 tests, but we still discuss with the company, and it's really possible that we will have during next week we will expand our production up to 40,000 tests. Do you know if the plasma of COVID cure patient can be used for drug clinical trials? I've heard about the one about the one trial only, where the plasma was used as the medication. I have nothing about the other attempts to do it. Sebastian asked, will you write a paper about this? Well, I don't know, actually. Maybe we will ask, because we collected many samples. We have plans about these samples, so hopefully we will publish something about the sequences, about the different things, but I don't know whether the kit optimization is the material for a paper. Is there any principal difference between tests from China and Poland? Yes, there are. There are many. Actually, there are many kits, as you might know. There are many kits, diagnostic kits, available in the Polish market. So they are from China. They are from Turkey. This is a very popular kit in our diagnostic units. They are from England. I've heard also about the kits from Taiwan. So what is the principal difference? The kit from China, for example, which I know, they do not contain the reference genes, which is human gene. So they do not prove the appropriate biological sample handling. So when we saw this a few weeks ago, we assumed that this kind of control has to be included in the test, because when you produce negative patients, many negative patients, and you know nothing about the biological samples of these patients, so you know nothing about the storage conditions, about the taking the samples at the beginning. You don't know whether the patient is negative because it's negative about in case of SARS-CoV-2, or it's negative because the material was prepared or was taken in an inappropriate way. So this is the main difference in our kit comparing to the kits, for example, from China. And also the other kits looked somehow in a complicated way. So I mean complicated way. They contain the more than 2 genes. For example, 3 genes, 2 for virus and 1 for so-called spike-in. And all of these genes are detected by the probes, which are labeled in 3 different fluorescent dyes. So it means that we need the machine which detect, or it's equipped with at least 3 channels. And this kind of machines are not really popular in. They are not as popular as the machines with 2 detection channel, which we have in the institute, and actually many institutes or universities or diagnostics unit work with. Okay, how long the results appear after have the testing? How long the results appear after? I'm afraid I can't understand the question. How long the results appear after have the testing? Gali, can you explain the question? Sorry, I can't understand it. Is the machine different from gel electrophoresis machine? Yes, the machine is totally different from electrophoresis machine, because the qPCR machine detects the quantity of the changes. So it detects the quantity of RNA from the virus and RNA also from the sample itself. So we are still waiting for the questions. Okay, so the question is about the time of the diagnosis. So we say as short as only possible. So from our experience, we've already diagnosed more than 4,000 patients. So we know that the time between the sample taking and the diagnosis has to be as short as only possible. But when you consider the diagnosis itself, so RNA isolation and the detection and the final results, with the handling with many samples as we do, it's 5 hours. Okay, thank you very much. I see the question from Matthew. Matthew, if you want to contact me, please write me an email. Okay. So do we still have? My email is present. You can find my email on the webpage of the institute, of our institute. So you can find it, Matthew. Okay, so I think, Michał, that's it. Can we finish? Okay, so guys, thank you very much for being with me. Hopefully, I said at the beginning I didn't waste your time. Thank you very much again.